Thank you, Noel, and thanks also to the two previous speakers, because both of their presentations, I think, will provide comparisons and contrast to what I'm about to speak to. In addition, I won't be presenting original data from trials that have recently been conducted. I'll mostly be going back and trying to put into context some earlier work, both from my lab and others, with respect to this overarching question, which is how much does the resistome change with age? Noelle, in all of her wisdom, gave me the title of association with age. I'm going to start, because this is what epidemiologists do, we machinate about the idea that association is not causation, but ultimately we're looking for causation. So what I'm really interested in is what are the effects of aging on the resistome? And you'll see, I hope, and you'll understand that it's actually a difficult question. So acknowledgments usually come at the end, but I want to start at the beginning, because on the left side in particular are individuals that we've been working with on an ongoing project to try to better understand this question. And I think in particular Victoria Volkova at Kansas State University deserves most of the credit for the ideas and the approaches that I'm going to speak to, but also other individuals who are at this conference, including one who's sitting right here, are duly acknowledged. I'll also present work from past colleagues when I was at Kansas State, as well as work at Texas A&M. So we need a bit of a glossary. I don't know if you're like me, but this omics thing it drives me nuts, because pretty much every week there's a new omics. But if you go to Wikipedia, which is my go-to source, and depending on the day, the definition, if it hasn't been edited, will suggest that it's a collective characterization and quantification of biological material. Okay, so you'll have transcriptomics, metabolomics, and so on. But the one we're mostly interested in today is something called genomics, which when I went through school was genetics, but it's more than that. Essentially looking at the structure, function, evolution mapping, and someone editing, the CRISPR-Cas folks, of genomes, which is an organism's complete collection of DNA. Now, for bacteria, that's not just what's in the chromosome. It extends to include plasmids and other mobile genetic elements. Metagenomics, I didn't like the Wikipedia definition. I think the environmental scientists had got at it the day that I looked at it, which referred instead, I like what the microbial ecologists describe, which is studies of the genomics of metapopulations. And for those of you who aren't familiar with the term, it comes from ecology, and it refers to what appears on the outside to be a fairly stable population, but in essence it isn't. So if you think about the city of St. Paul, Minnesota, people can say the population is, I don't know what it is, 1 million, maybe less. But as you know, in the period from when we woke up this morning to the present, some people have died, sadly. Others were born. Some have moved away, some have moved in. And so it's a population, but it's not always the same people. And if you think about the feces, for example, it's clearly not always the same bacteria. There's a certain flow-through effect that we're all familiar with. But the same applies to the oral cavity, the nares, the skin. And so the metapopulation is the genomics of the whole of metagenomics is the study of the metapopulation. There are a variety of methods. I'm going to speak only to two of them today. 16S rRNA metagenomics, which tries to type the bacteria that are present in the population, and shotgun metagenomics and various sort of hybrids thereof that try to describe the genetic material that's present regardless of whether it's bacteria or other targets. So the resistome, and this was again the title that was given to me, but the resistome has actually not got a very good stub in Wikipedia. It's a fairly recently coined phrase attributed to Gerard Wright, at McMaster University, and several people have sort of taken it and run with it. And one of them is Gautam Dantas, another is Morton Summer, and actually 10 years ago, to this date almost, their paper appeared in Science, which was one of the first to sort of define the metagenome in the microbiome of their own stool relating to beta-lactamases. So it's a fairly new field, but the idea is that all of the resistant genes and their precursors, both in pathogenic and non-pathogenic bacteria, pose a potential risk to health should those genes find their way into pathogenic bacteria in particular. So if you study the microbiome, there's always requisite graphics that you have to present. And one of them is this principal component analysis on the right side. But I think if we look at what do we know about old versus new in terms of the microbiome, one thing that's clear is that when animals are first born, they have a relatively less diverse population of bacteria inhabiting, in this case, their intestinal contents. And some of that is simply a lack of access to acquisition of a number of bacteria, so they're exposed to the sow in the case of piglets, or babies to their mothers, both through the birth process, but also on the skin and the environment. And so if you measure it overall, nursed versus weaned piglets have less diversity, and that probably doesn't surprise any of you in terms of the diversity of the organisms. In terms of the principal component analysis and the diversity, that's also illustrated over here. But in some ways, you actually can see that depending on the axes that you look at, it isn't always as consistent. The other one is that bacteria come and go. So when piglets are nursing, they're acquiring a diet of milk. And when they're weaned, the milk goes away, and there's a substitution. And in particular, the substitution that we're interested in is the Enterobacteriaceae become a much lower component of the total gut. So that includes E. coli, Salmonella, and others. And they're replaced by other bacteria that we would generally see as more favorable to the health of these pigs. So some good things happen in terms of the potential for bacteria, but these don't go away entirely, or we wouldn't be able to see studies like Clarissa's on E. coli, obviously. So let's switch. You won't see another one of those graphics now. Let's switch in from pigs to cattle. And the reason I'm doing this is to sort of illustrate to you what you can see if you look at tables and you look beyond what the authors might be trying to present to you. So this is the simplest example I have. A dairy calf in a hutch. If you look at its umbilicus, it's actually fairly recently born. Obviously, its diet depends on what's placed in those buckets, but it's also exposed to everything in its environment. This obviously is not the same coloration, so this is not the adult version of the earlier one, but this is the adult lactating dairy cow producing milk. And so we asked the question, what are the differences that are attributed to aging between those two groups? And again, we'll talk about associations because we can't talk about causation. But I'm drawn to this table that I first saw in 2005, so almost 15 years ago, by Kenji Sato, who's since passed away. When he was a student at Michigan State University, he was on a project looking and comparing E. coli resistance in organic versus conventional dairy farms in the Midwest. And these happen to all be from the neighboring state of Wisconsin. I'll draw your attention to the column, which represents the number of antibiotics to which the isolates were resistant, similar to what Clarissa presented. And they range from 0, which is pan-susceptible, through 12, which is the total they tested. These would be considered pan-resistant. The columns as presented are cows, calves, conventional, and organic. And you'll notice that the total is roughly 280 E. coli isolates for each of those four groups. And the authors, of course, were drawn to comparing conventional and organic, and so they described non-significant differences between conventional and organic farms in terms of the multidrug-resistant profile. So essentially no differences. And most of them were pan-susceptible with relatively few single, double, and triple resistances. They then compared calves, conventional and organic. And the interesting thing is here that, in fact, now they found that conventional calves had fewer pan-susceptible than organic and more, if you will, single, double, and multidrug-resistant profiles. And so they noted that the p-value less than 0.001 suggested significant differences between organic and conventional. What drew my attention was this arrow, which they don't talk about. And that is the calves to the cows. And if you present the data instead by turning them on the side, the x-axis is the multidrug-resistant profile. We see that among adults here, we see a high peak of pan-susceptibles. And the green boxes essentially sit around what would be classed by CDC as multidrug-resistant, resistant to 3 or more classes of antibiotics. If you compare across conventional if you look at conventional dairy cows versus conventional adult cows, you see much higher levels of this. If you look at organic to adult, you see much higher levels. But most importantly, if you look at the young versus the old, these being the cows versus the calves, regardless of whether they were raised with antibiotics or not, you see that the calves had much higher levels of antibiotic resistant among the E. coli than the cows do. And this is inexplicable. We would like to say, well, it's the antibiotic exposure, but we know that the organic calves, although there are some rules about transitioning to organic, but let's assume that there's less exposure in those. Clearly, that difference has nothing to do with that and is more likely to be something associated with aging, if not aging itself. So we can accept that. Then we ask, well, what about pigs? And I thought it was interesting that the comment about ceftiofur came out. Because not so long ago, we were doing a study in a recently weaned pigs sourced from farms in the Midwest. And we were using quantitative PCR to estimate the load of this particular gene known as BLA-CMY2, which is a cephamycinase that confers resistance to third-generation cephalosporins. And the piglets, or the pigs, I should say, that we were acquiring came in with roughly 10 to the power 6, or 1 million genes per milliliter of feces, or gram of feces. And we were applying these treatments, a control, chlorotetracycline at therapeutic doses, copper, and a combination. And we were really excited because these are smooth curves, but we saw an initial drop in the levels of resistance in the copper CTC group that suggested it might have an effect on reducing resistance. Later, they all essentially leveled out, and so by the time the study ended, we saw no differences. The important thing here, though, is not the treatment groups. The important thing is here is why did it go from 10 to the power 6, which is roughly 1 million, down to a little over 10,000 over a space of 5 to 6 weeks. And again, one can only imagine that aging had something to do with this. However, we also have to recognize that several different things occurred. We purchased the pigs, they were transported, they were placed in a new environment, they were placed on a new diet, and so it's essentially impossible to separate out the effects of aging versus the changes that we just described. So epidemiologists spend a lot of time wringing their hands about association versus causation. The other thing we obsess about is this idea of studying 3 things at one time when one of them is closely related to the other 2. And so the classic conundrum of the age period cohort effect is illustrated as follows. Here you see 1920, 50, 80, 2010, 2040, 2070. These are separated into 30-year blocks, and the idea is that we have young, middle-aged, and old individuals, humans. So if this is a cohort, we have the young, the middle-aged, and the old, and we have separate cohorts, birth cohorts, born in those 30-day periods. Most studies that we know about, especially those that are funded by the federal government that last less than 5 years, will look across these cohorts to determine associations between age and hypertension, age and certain disease outcomes. But the problem is that you are comparing groups from different birth cohorts. And you might say, well, so what? Right? You know, we know that older people have a higher risk of a certain disease, but do you really know that? Because what you're looking at when you're looking at this group is a group that was a part of this. This is the middle-aged group. In 1980, they were part of the young group in 1920. But you can't necessarily assume that we can separate out those cohort effects with the period. Okay? And if you don't believe me, look at this graphic, which is, we could spend the entire seminar talking about this, and I'm not going to do that to you. But suffice it to say, each of these lines represents life expectancy for at birth, 1, 5, 10, 20, 30, 40, 50, 60, and finally 70-year-olds. Okay? And you notice that a 70-year-old might live to 75 back in 1850, and in 1950 they might live till 75. On the other hand, a 1-year-old back in 1850 might live to 50, versus in 1950 they might live until 70. So things have improved a lot for those who were born recently versus a long time ago, but they've only improved for those who were part of the younger birth groups. And what happens over time is we have events like the First World War and the Spanish flu and the Second World War that really mess up cohorts in terms of who survives these major events. Now, we're talking about bacteria here. We're not talking about people, but the concepts are the same. Because if we don't examine the cohort and the age, or the period in the age, or the period in the cohort, we won't really understand necessarily what's going on through time. So this illustration, for those of you who didn't spend time in genetics classes in the 1980s trying to keep Drosophila or fruit fly colonies alive to breed them, will understand, let's talk long term. Where do you see yourself in 5 days? The idea of the counterfactual is the idea of a life experienced, a life not experienced. So when we apply treatments to groups of pigs, for example, and we assign pigs to groups to receive them, we're doing that simply because we can't have a pig experience both treatments. Right? It's counter to fact. They're either assigned to one or they're assigned to another. But in an ideal world, another world, we might be able to experience both at the same time. Okay? And so these non-received treatments are known as the counterfactual treatments. And they're important philosophically because they don't exist, but they allow us to imagine a scenario in which confounding and collinearity are not important. So the counterfactual or potential outcome would be the pig that received Draxin, penicillin, tetracycline, and control. It received only one of those, and the counterfactuals are those that it did not. So how did it respond if it had received the others? You may say, you're crazy. It doesn't happen. But it's actually useful for conceiving of this problem. Okay? Now, if we identify factors that are closely associated with aging, okay, through time, and we attempt to account for those in explaining observed phenomena, we can come up with alternate explanations that actually offer an opportunity to intervene. Because there's one thing that we know, is that there is no counterfactual for aging, not one that we can imagine in this reality. So imagine an individual who doesn't age. Can we generate a counterfactual to determine the effect of aging? No, we cannot. So what do we do instead? We identify things that are associated with aging, such as birth, weaning, changes in diet, changes in environment, and so on. Taking us back to this imaginary study, which wasn't, when these piglets arrived with really high loads of beta-lactamase resistance in their feces, what did we know happened when they arrived? Well, the environment changed. There was a change in feeding ingredients. There was a change in the antibiotic regimen. What we don't know is what the counterfactual group that remained at the source that may have aged similarly in terms of what they experienced. So I'm going to challenge some dogma here, and it's not meant to offend any of you. But I'm going to say some things that I believe are based in fact, but that may run counter to what you all believe. I can show you from work we've done where we follow animals closely immediately after they're treated with antibiotics that there's a profound effect of the antibiotic in selecting. The blue line represents susceptible E. coli. The red line represents resistant E. coli. And these are dairy cows that receive a double dose of Exceed or crystalline cephtia-free acid. This is a single dose in a group of beef cattle, and the phenomenon is the same. We drive down levels of susceptible bacteria in the first few days, and then it recovers slowly over time, which allows resistant populations to fill in the niche, which eventually dissipate. Okay? We can measure immediate effects dynamically. However, if we take a sample on day 0 and we take a sample out at day 60, well after this trial ended, we can see no differences. We can see no effect of the treatment. And so many people would say there's no effect of the antibiotic in affecting resistance in that population. I can show you it dynamically here using indicator bacteria and selective approaches. But in the short and medium term, antibiotic use and resistance appear to become decoupled. Okay? In the long term, however, as these effects accumulate over time in individuals, in herds, and across herds, we actually see a change. And I'll show you how here. This is a rather pixelated image from a student dissertation. They obviously didn't use sufficient graphics or DPIs. This is one dairy farm, a second, and a third. The red line are cows that calve that didn't have metritis. The blue line represents cows that had metritis, were given a double dose of Exceed. And you'll see that the drop, which represents the difference in log 10, which is a bit problematic if you're not familiar with CFUs and logs, But suffice it to say, the lower this number that gets, the higher the proportion of resistant bacteria at that point in time. In dairy 1 out of 32 of the bacteria in the feces at that point are resistant to a third-generation cephalosporin. In dairy 2, 1 out of 316. And on dairy 3, 1 out of 3. Okay? And these are measurable and significant differences. The phenomena looks the same, but the drop to this endpoint, or the highest peak, is dramatically different among the farms. And the differences we can show from other data not shown are the historical levels of use of this product. In fact, if you look carefully, you'll also notice that the starting values are about 1 log lower in this dairy than in the others. So it matters in terms of the herd and the history of the antibiotic use, what the effect you're going to see is, which sort of comes down to the replication problems that were earlier described. The other thing I'll show you, and again, is this idea that aging is associated with other phenomena, is we have seen several times, unfortunately these are still unpublished data, but these were recently published in the Journal of Animal Science, that if we take calves off grass in extensive environments, place them in feed yards, they have essentially zero resistance to macrolides, lyncosamides, and streptogramins. This was repeated recently in an entirely different feed yard 10 years later. We see the same phenomenon. They come in off grass with no resistance, and then by the time they're reimplanted, or 90 or whatever days later, suddenly we see high levels of resistance. And it doesn't matter whether they got the antibiotic or not. Okay? In this study, it was tylosin, monensin, or a combination, and the control group is in dark gray. In this study, the control group turns out to be the highest, actually, in terms of erythromycin resistance. So what this tells us is that the change in the environment, potentially the change in feed, but certainly a decoupling of antibiotics, indicates that antibiotic use, at least in the short and medium term, has very little effect. And you could argue that 118 days also has a potential aging effect, but I'm hoping to convince you here that at least in the context of a post-ruminant world, that isn't the case. So, I believe that aggregations of use are most important. Using an antibiotic in an animal or a person or a group selects for antibiotic-resistant bacteria. They can spread to other individuals and groups. And initial AMR fitness disadvantages disappear over time. The prevalence and the quantity gradually increases. And if we use bacteria like E. coli or Enterococcus like Clarissa used, you can see this. Salmonella is more problematic because there's a distinct serotype resistance linkage. But these are data that are getting a bit dated, and I'd like to replace them, but we haven't had a new antibiotic class since 1987 in agriculture, so they still fit. We see relatively low levels of resistance, at least none detectable in the first decade, and then we see a gradual rise. And the question is that a response to the use of antibiotics in any particular animal? No, this is a change in the background level, which we saw in dairies 1, 2, and 3. Obviously, it's not equal across all species. I showed you cattle. This is pigs that didn't experience the same response. And again, largely because the salmonella that are found in pigs more commonly than in cattle didn't have the BLA-CMY2 gene at the time. But this phenomenon is going on in human health. Okay, it's well documented that since the new millennium, there's been a reasonably elevated rise in cephalosporins, third-generation cephalosporins. Fluoroquinolones, these are critically important. You can see that the aminoglycosides project back a lot further because they were introduced far before that. But now we're seeing carbapenems and recently colistin resistance. And the increase in the levels of these is not in response to use in the immediate sense. It's a cumulative effect of the accumulation of resistant materials. So, what can we do about it? I've already given you the doomsday scenario about the cumulative use, the suggestion that what happens is what we start with affects what we see in those dairy farms. This paper came out in Science in 2013, and the Jaccard Index is a sense of the relatedness of the population. This is the only study I'm aware of that actually had funding to go beyond 5 years in a human population. So they resampled the same individuals, some of whom who were taking a weight reduction diet, a very bland diet. And over 5 years, you could see that there's a very strong retention of the similarity of the fecal microbiome. Interestingly, it's less so in those who lost a lot of weight, okay? But down here, I won't ask you to look on the right side, you'll see a pattern, which is a large peak over here on the left, which is at the family level, the genera, the species, and the strain. You see very low levels of similarity in terms of individuals versus others that were not themselves. If you look at the same individuals versus themselves at the same time point, you'll notice there's also a peak out here in terms of strain, and that is sustained within an individual over time, which suggests and supports the idea that the microbiome that you have now is reasonably stable over time, in this case up to 6 years, but it's different in many ways, at least at the strain level, from other individuals in the room. And that might make you a little more content, I don't know. Obviously, at the family level and the genera level, we see similar patterns, although they're much, much closer together. Now, this is important because there's a great deal of interest ever since the discovery that fecal transplantation helps patients with Clostridium difficile or Clostridialis difficile dysbiosis. In fact, the scores for the use of that treatment are off the map in terms of its efficacy. But these individuals who have dysbiosis have a highly disturbed gut that's been taken over by a toxigenic bacterium that essentially has changed the gut, allowing sort of a new niche. But there are values potentially in looking at adding new types to microbiomes. Okay? What if we had a highly resistant microbiome and we'd like to substitute susceptible bacteria for those, perhaps in animals going to slaughter? Is that easy to do? Well, you'll notice that recently people started talking about the microbiome as an additional body organ, and so the concept of not transplantation but engraftment is now sort of in vogue. So if you can accept the idea of grafting new organisms in the microbiome, the question is, how could it take place? Well, you would introduce it, it would get established, it would grow and persist, and then it might impact the other bacteria through competition and antagonism. Okay? The problem with this is it's exceedingly difficult to do. All apologies to the probiotic or DFM industry is if the idea is replacing existing organisms as opposed to maybe affecting some other health-related change, it isn't that easy to do. And you saw that in the previous persistence talk. I came across this paper actually recently as I was putting this talk together, and I find it fascinating. Andreas Bäumler, who's the lead author, and his postdoc have proposed something called the Houndour Hypothesis. And it's adding on to the nutrient niche hypothesis that helps explain how microbiota change over time. So the nutrient niche basically says if there is a substrate present necessary for a bacterium, that bacterium can exist or could be introduced. But if you take that substrate away, the bacterium will disappear. And the example they give is of Bifidobacterium infantis, which in human breast milk provides oligosaccharides necessary for this bacterium to survive and thrive. Once the child is weaned, the bacterium disappears. I think it reappears in various yogurt products, but it disappears during this period. Okay, the idea of prebiotics feeds the idea of the nutrient niche hypothesis. However, probiotics don't necessarily work unless they work as a Houndour hypothesis. So in Bäumler's paper, they basically suggest that either one, if you tell the blue pill or the red pill, the bacterium that's first introduced will occupy a nutrient niche, and it will provide resistance, if you will, colonization resistance should you try to cross-introduce them. The only way you can achieve it is potentially to disrupt that or provide additional niche. So how would we do that? How are we doing in time? 20 minutes. Okay. In poultry, we can look at the hen, the hatchery, or the feed environment. In pigs, we can look at feed. We can look at, obviously, farrowing to weaning and growing. In cattle, we have the additional advantage of extensive versus intensive phases in terms of beef production, cow milk, monogastric ruminant. And in dairy calves, which I obviously used examples of, we have monogastric and ruminant. So I have some closing remarks. First is the shifts in microbiome and resistome that are associated with aging are notoriously difficult to understand or manipulate. The second, if you consider antibiotic use as a primary driver resistance, it doesn't make sense in terms of neonates versus adults, in terms of the quantity of resistance. Founder and nutrient niche theories offer an explanation and opportunity to modify these, and probiotics sort of feed this idea. However, if resistance is decoupled in the short and medium term, they do recouple in the aggregate form. And the historical and cumulative use of antibiotics appears to be the biggest driver for what we see in the present day. For example, look at tetracycline resistance and carbapenem resistance. My last point is the one that worries me the most. The abundance of resistance genes is lower in adults than in infants and neonates. However, the diversity of the resistome is not, which means the potential for selection out of the resistome remains great in adults, even if the abundance is not. There's a recent report in Mortality and Morbidity Weekly Reports from the CDC of a Salmonella Newport outbreak. Mexican queso is under suspicion, but so are cows raised in the Texas Panhandle that have been found at slaughter. This organism is resistant or has reduced susceptibility to azithromycin, a macrolide, and ciprofloxacin, in addition to others. Neither to lathromycin, which is a azithromycin analog, or enrofloxacin are approved for use in adult dairy calves, but they are approved and anecdotally used a lot in calf rearing. Co-selection with ceftiofur is a possibility, and we've discovered this. If we use selective media in those 3 dairy farms, we identify ESBL or CTXM variants in these all 3 dairy farms, CTX1, 15, 32, 27, 55, 65. But we also find in 2/3 of the case Q and R genes, which are quinolone resistance, and MPHA, which is the gene found in this Salmonella Newport. Worrying to us is the idea that we're carrying over from the resistome of the calves to the adults. It gets suppressed. It's hard to detect, but it's there. And so the effect of selecting in the calves may, in fact, remain as a potential risk in adults who, if a linked antibiotic such as ceftiofur, which is very popular in dairy production, is used, it actually helps pull it out of the background and create a new risk. So what are the effects of aging? I'm not going to say there aren't any, but this is the unanswered question. Okay? So I can't tell you there's no effects of aging. You can see it for yourself. You can see it in me. The real question is, how do we create a counterfactual that actually allows us to assess the effects of aging on things as complex as the microbiota rather than phenotypes like hair?